intrinsic pathway, which is also called as a caspase dependent pathway. In this intrinsic pathway, there is some intrinsic reason is there, that is a cell death is there, or a intracellular stimulus is there, like any DNA damage is there, or cell undergoing hypoxia, or there may be generation of the free radicals. So because of this, it doesn't require any extracellular stimulus, because the stimulus is from the inside the cell. Therefore, it doesn't require any receptor on the surface for receiving that stimulus. In case of extrinsic pathway, it is a death triggered signal from the extracellular, that is outside the cell. So it requires a receptor for receiving that signal from outside. So reason for the intrinsic pathway: intracellular death signals are there, like DNA damage, or there may be generation of the ROS, that is reactive oxygen species. Then some oncogenic stress is there, that is some abnormal condition in the body that can lead to the oncogenesis, that is cancer, or there may be any viral attack or lack of the growth factors. So these are the intrinsic reasons or internal reasons are there for stimulus of the apoptosis pathway, intrinsic apoptosis pathway. So this intrinsic pathway, it is initiated within the cell. Intrinsic pathway, it uses a mitochondria as a central component for activation of the apoptosis. So this is the pathway. In this, there is the apoptic signal is there, and this apoptic signal, it lead to activation of the protein, that is P53. P53, it is an important gene in the body that will lead to a apoptosis. Then it will activate a back and Bax gene. Usually in the normal cell, these back and Bax genes, they are controlled by another proteins, that is BCL2, BCLXL and MCA1. So this BCL2 and BCLXL, they are the anti-apoptic proteins, and back and Bax, they are the pro-apoptic proteins. They are called as a pro-apoptic proteins because they stimulates the apoptosis, while BCL2 and BCLXL, they are called as anti-apoptic proteins as they inhibits the apoptosis. So in normal condition, these back and Bax, they are bound by the BCLXL and BCL2. When there is the apoptic signal in the body, that is any internal abnormal condition is created in the body, then it will lead to the inhibition or degradation of these proteins BCL2 and BCLXL. So because of degradation of these anti-apoptic proteins BCL2 and BCLXL, it will this Bax and Bac, they are remain free, and this free Bac and Bax protein, it will lead to formation of the pores to the mitochondrial membrane. As the pores are created to the mitochondrial membrane, there is a release of the important component, that is cytochrome C. This is a very important component which is involved in this intrinsic pathway. Cytochrome C, then it will lead to a formation of another factor, that is apoptosome. It will lead to a activation of the APAF1. APAF1 is the apoptosis promoting factor 1. This cytochrome C bind to the APAF1 structure and forms a wheel-like structure which is called as a apoptosome. Apoptosome has ability to activate caspase 9. Initially it is a procaspase 9, which is an initiator caspase. Caspase 9 is formed by the proteolytic cleavage of the procaspase 9. Then caspase 9 then activates the caspase 3 and caspase 7, which is a executioner caspase or effector caspase. This caspase 3 then lead to degradation of the nucleic acid. Its role is to degrade the inhibitors of the nuclease enzyme. As the nuclease inhibitors are degraded, nuclease enzyme is free to degrade the nucleic acid, and this will lead to the death of the cell, that is apoptosis. So this is about the pathway of the intrinsic pathway of the apoptosis. So in unstimulated state, these cytochrome C, it is trapped in the inner mitochondrial membrane. As the intrinsic stimulus is there, this cytochrome C, initially it is in interaction with the lipid cardiolipin. But as the stimulus is there, it will releases out. The cytochrome C, it is an important component in the intrinsic pathway. It is a water soluble component in the mitochondrial electron transport chain. And here the main role of the proteins BAX and BAC is there, which is a pro-apoptic proteins. That is it is apoptosis promoting factors. In normal condition, these BAC and BACs, they are regulated by anti-apoptic proteins, that is BCL2 and BCLXL. So the steps involved in the apoptosis, in this the first step is the internal DNA damage signal is there. For example, DNA damage is there, or any hypoxia, or generation of the free radicals. So that might be the internal signals are there. This is the summary of the apoptosis or intrinsic pathway of the apoptosis. So we will see that steps one by one. First step is the internal DNA damage. Then the another is inhibition of the anti-apoptic proteins is there, that is the BCL2 and BCLXL. So these anti-apoptic proteins BCL2 and BCLXL, they bound to this BAX and BAC. That is, BAC and BACs are regulated by that anti-apoptic proteins in normal condition. But whenever there is any internal DNA damage is there or apoptic signal is there, then it will lead to degradation of that anti-apoptic proteins. And because of that, the apoptic proteins or pro-apoptic proteins like BAC and BACs, they free. And the free BAC and BACs, it will lead to a oligomerization of the BAC and BACs, and it will create a pores to the it will create pores to the mitochondrial membrane. It will create pores to the mitochondrial membrane, and through that pores, cytochrome C releases out. Cytochrome C, it is initially present in the internal membrane of the mitochondria. It releases from internal membrane to the outer membrane. Then after this, there is the activation of the factor, that is APAF1, that is apoptosis promoting factor 1. As there is the activation of this APAF1, APAF1 get bound by cytochrome C molecules, and there is the formation of structure which is a wheel-like called as a apoptosome. Then apoptosome formation is there. After that, this apoptosome, it will lead to activation of the caspase 9, which is initially pro-caspase 9. It will lead to a formation of the caspase 9, and after that it generates a cascade. There is the generation of the caspase 3 or activation of the caspase 3 is there. Caspase 3 then lead to inhibition of the nucleic acid, as its action is to degrade the nuclease inhibitors. As the inhibitors of the nuclease enzyme, they get degraded, nuclease enzyme is free to degrade the nucleic acid, and finally there is the cell death. So this is about the process of intrinsic pathway of apoptosis. So extrinsic pathway of the apoptosis we will see in upcoming videos. Thank you.